Good morning. Today I would like to discuss automated DNA sequencing. Automated DNA sequencing. Now we can automate DNA sequencing. This automated DNA sequencing have been developed to sequence a really large amount of DNA. Okay, the automated DNA sequencing has been developed to sequence a really large amount of DNA. Okay, that means when we do the DNA sequencing using basic Sanger's method of sequencing. Okay, Sanger's method of sequencing. That time we can only do the small amount of DNA. Okay, small amount of DNA because this band separation will do by manually, and that's why we can do only very small amount of DNA sequencing is possible only in the case of basic Sanger's method of sequencing, and but in the case of automated DNA sequencing, it is possible to sequence really large amount of DNA. And the principle of Sanger's method and the automated DNA sequencing is the same. The principle behind the automated DNA sequencing and Sanger's method of sequencing, they're same. Okay, same. Okay, that means the chain termination PCR is used in automated DNA sequencing and the Sanger's method of basic Sanger's method of sequencing. Okay, and the primer extension. Okay, the amplification primer extension also is same like as a basic Sanger's method of sequencing. Okay, okay, this is the primer extension reaction also is the same as like basic Sanger's method of sequencing. And in this automated DNA sequencing, we'll use DNA polymerase, DNTPs, ddNTPs, primer that will use in the same case of the in the automated DNA sequencing also. Okay, but the difference is that the ddNTPs, okay, the ddNTPs, that means chain terminator ddNTP nucleotides, that will be fluorescently tagged in the case of automated DNA sequencing. Okay, the ddNTPs is fluorescently tagged. Okay, see this ddNTPs are fluorescently sequenced in the case of fluorescently tagged in the case of automated DNA sequencing, and the primer will be unlabeled. Okay, the primer will be unlabeled. In the case of Sanger's method of sequencing, the ddNTPs will be uh, not labeled, but the primer will be labeled in the case of Sanger's method. But here, this ddNTPs will be fluorescently tagged, and that fluorescent color will be different. For example, you see ddTTPs. Okay, ddTTPs are here marked with the red fluorescent, and here TC is marked with the ddCTP. Okay, dideoxy CTP is marked with the blue, and here is DDATP is marked with the green, and the DDGTP is marked with the violet color. So different colors marker were used to mark the fluorescent tag, or in the case of DDNTPs. Okay, DDNTPs. Okay, DDNTPs. You see, DNTPs is there, and DDNTPs also is there. Okay, DNTPs also is there, and DDNTPs also is there, and there's a difference. And one more difference is that in the case of Sanger's methoding, we are doing the reactions in four separate tubes. Okay, we are doing this amplification in four separate tubes, but in this case, we'll do all reactions in a single tube. That means, for in the case of Sanger's method of sequencing, for we are using TTP, we'll use one tube, and for DDCTP we'll use second tube, and we'll use DDATP for that we'll use the another tube, and DDGTP will use another tube. But in the case of automated DNA sequencing, we are doing all procedures in a single tube. Okay, all procedures in a single tube. That means a reaction carried out in one tube, and after that amplification. Okay, we'll go for the amplification after adding the all components, enzymes, template DNA, primer, DNTPs, DDNTPs, and we'll get the fragment with varying length, like is we obtain in the 4 tubes of Sanger's method, but here we'll get the all fragments in a single tube. Okay, after that, after the reaction, we'll go for that reaction products will undergo for separation through the capillary electrophoresis. Okay, capillary electrophoresis, in that case we'll separate according to size. That's done in the case of a Sanger's method or any electrophoresis. We can get the different products by that capillary electrophoresis, and we'll get the different fragments. Okay, and different fragments will get, and in that case that will that's we told that the DNDP are fluorescently tagged. Okay, and then after separation, this electro will, when we pass the laser through this band, okay, laser light through this gel, that time this fluorescently tagged DDNTPs that will terminate that has terminated the DNA fragment, that will observe as a band in a capillary electrophoresis tube. Okay, capillary electrophoresis tube. Here you can see, here you can see we have a primary is there, a template template DNA is there, template DNA is there, primary is there, we added DNTPs, DDNTPs, and that enzyme is there, everything is there, and we go for the reaction time, and the time you see here, when after that here one strand is terminated, that time when water strand is another strand is that is also terminated here after adding two nucleotides, here also adding. But in the case here we told that all reaction is carried out in a single tube, and we'll load this amplified product in a capillary for a capillary electrophoresis, and when we pass this laser light, okay, when we'll pass the laser light into this capillary electrophoresis gel, okay, capillary electrophoresis tube, that time when laser is passed through this fluorescent tag will be produce fluorescent light, and that will be detected by the detector. Okay, that will be detected by this detector. For example, when we pass from here, you see the here is the violet color is there. So, okay, violet color is given here, so we can see which DDNTP is marked with violet. So here when you come here, the violet is the DGTP. So we can see that, say that here the first nucleotide, that is a guanine, and here you see, and second also that given the violet color. Okay, so when we the light pass through this, that produce the violet color, so the detector will detect the color of that, and depends on that color the system will generate a chromatogram. Okay, system will generate a chromatogram. In that we can see different fragments here, different peaks here. You see here two violet peaks here. That means that G is there. Okay, 2G is there, and after that you see red peak is there. So red means, red tag means here red tag is there. So red tag means T. Okay, T is there, and here you see here blue is there. So come here, the blue tag means that C. Okay, here C is the C. Like that we can identify the sequence. Okay, we can identify the sequence, and the band, the color of band we observe here is depends on the which DDNTP is terminated the synthesis of this fragment. Okay, which DDNTP is terminated the synthesis of DNA fragment, so that fragment will give that the DDNTP's fluorescent color. So here DDNTP guanine is terminated here, so it's given violet color, when laser light is passed through this. And here also violet color, so DD GTP, okay, DDGTP is a violet, is a DDGTP is here. And here you see that here you see red is the, okay, red is the, red means red when laser pass through this, and then you in the red color, so we can detect, this will detect red is a, here see red is a. And here you see which one it is a, this here blue color is there, okay, blue color, blue here come here, and blue is the C, okay, like that this system will detect this, detector will detect the color, and from that color this software will generate a chromatogram, in that we can clearly isolate, identify which is a sequence of this DNA. Okay, this is about the, okay, this is the automated DNA sequencing. I hope you understood this. Thank you for listening. So thank you for listening.